Thank you, everybody, for joining in. And I know we're going to have more join in as we go along, but we have a special treat today, and I'm really excited about it. And I'm going to turn this over to Dr. Tom Gill, who is the chair of the Smith International Center, and he's going to be introducing our special guest today. Tom?
Great. Thanks, Deb. Good to see everyone today. Thank you for joining us. We have the privilege of having Dr. Elaine Dorneles from the Federal University of Lavras in Minas Gerais State, Brazil, today. I met Elaine earlier this year in February. I was there with Dr. Hongwei Shin, our Dean for Ag Research, and Dr. Bruno Pedreira, who is faculty in plant sciences doing forage work. And UFLA is one of Bruno's alma maters, so we were there and making great connections. And we have put in for some internal funding. So we have some funding from the Office of Research here at UTK to be able to build on our Brazilian collaborations. And so this is one part of that where we are having different scientists speak with different units across our campus. And so this is one which we wanted to do together with One Health. And then we are bringing those scientists here in the spring. So we'll be doing a week where we have a workshop and have those scientists visit with different faculty and staff and students and see things across the state. That'll be the last week in April. So be on the lookout for more information about that. And so Dr. Dorneles will be coming to visit us in the spring. So here's a good chance to get to know her some and get to know her work as well. She is a faculty member, a professor in the Department of Veterinary Medicine at Lavras, and she leads work on infectious diseases of livestock, in particularly looking at bovine mastitis, AMR, use resistance, epidemiological modeling. And she looks at combining all of those things, how you combine the microbiological, the molecular, the data-driven approaches, all using that One Health lens. So, Elaine, over to you.
Oh, thank you so much. It's an honor for me being here and having the opportunity to share a little bit of what we have been doing here at UFLA with you. So I'm feeling privileged about having such a select audience to hearing me this afternoon. So I'm going to talk a little bit for antimicrobial use and resistance in dairy production, and especially about the challenge and strategy that we are facing here in Brazil, in Minas Gerais, and about our initiative doing it in a One Health approach. So Tom already introduced me, so I'm going to pass the slides. It's just saying that I'm a DVM, and I have my master and a PhD from UFMG and some part in Virginia Tech. And also now since 2015, and I'm a professor here at UFLA. So I divide the presentation. First of all, I will contextualize the animal production and antimicrobial use and antimicrobial resistance, just to put us all in the same background. And then I'm gonna talk a little bit about our project. We started last year a project trying to understand the antimicrobial use and thereby the resistance in dairy herds in Minas Gerais. We have some preliminary results I'm gonna share with you, and then my final considerations. So the animal produces growing fast in the last years. In 2015, in this year, we have a market that is the size of $5,000 billion, and we have expectation of growing, and will grow in the text of 6.2% until 2019. So this is the size of the market that we are talking about. And this animal production, we can divide in meat production. So I have brought here a map that show which are the big players in the animal meat production in the world, in the meat production. And we see China, Brazil, and US as the main players. Here, the data are counting for cattle, chicken, and sheep, all together pig meat. So we see that we are the bigger players in the meat production, and we have to say that Brazil is also the bigger exporter. of meat, all kinds of meat. So we not produce meat for ourselves, but for the international community. And here's the rank. Brazil is the third one in the meat production globally. And for the milk as well, we are in Brazil as well as you in US, big players. Brazil is the fifth one in milk production, but on the contrary of the meat, we produce milk mostly for our intern market. We do not export a great amount of milk. India is the first one in the milk production. US is the second one, and we have China also playing a very nice position here. And talking about Brazil, the dairy production is mainly concentrated in the southeast part of Brazil, especially in Minas Gerais state, the state I came from and where I live. And we respond for 1/3 of the Brazilian milk producer in Minas Gerais. So we have about 22 million heads of cattle here in Minas Gerais, and we produce, as I said, 1/3 of the milk in Brazil. It's about 9 billion liters per year that we produce. So here is Minas Gerais. We are in the southeast region of Brazil. And associated with the biggest market of animal production, milk and meat, we have the same countries figuring as the main consumer of antimicrobials, veterinary antimicrobials. We have China leading this rank, but also US and Brazil, they are the second and the third countries in the consumption of veterinary antimicrobials. So we have a very big concern, big concern about how animal production can influence or can have an impact in human medicine because of this huge consumption of antimicrobials. Since several antimicrobials that are used in animal production are the same that are used for treat human disease. So adjusting populations and weight, we can see that we consume 1.8 times more antimicrobials in animals than in humans. And 73% of the drugs, the bases, are shared between animal and human medicine. So most of the antimicrobials are not different. So everything we are doing in animal production can have an impact on the bacteria, especially bacteria that can cause disease, zoonosis in the human population. And also, when we use antimicrobials for animal production, milk and meat, we cannot have the full control about the antimicrobial that may be in the environment, especially here in Brazil, because even we have a very new legislation about how to do what the garbage in the rural areas. We do not have a way to collect the bottles of antimicrobials, so we usually don't know what happened with this antimicrobial that gets in the environment. So we have a bigger problem because we are impacting not only the human health, but also the environmental health. And also for wildlife animals, this can have an impact that we neglected completely. So here I brought some data that show that the amount of antimicrobials that is an average that we consume per cow in a year is 7 milligrams per kilo. And also the impact that we have today of the antimicrobial resistance infections in humans. We have a projection that shows that about 1.9 million deaths in 2050 will be because of antimicrobial resistance, because of bacteria that are resistant to all known antimicrobials. So this is scary, very scary and alarming. And I think people who work with animal production should pay attention and should do something about it. And I think in Brazil, the situation is the same that we see worldwide. In 2019, we have an estimate that in Brazil, 33,000 deaths were directly because of antimicrobial resistance, and 1,037 deaths were associated at least to antimicrobial resistance. And we know for sure, and because of the data that we have available in Brazil, that antimicrobial resistance kills more people than diabetes or kidney disease or respiratory infections or tuberculosis individually. So we have much more deaths that can be attributed to the antimicrobial resistance in Brazil than several diseases. And I got an estimate from US that you guys may spend $55 billion in healthcare in US because of the antimicrobial resistance. So which are our challenges here in Brazil? I'm going to tell a little bit what the problems we have mostly here and how we think about addressing this challenge. We have few tools nowadays to control the use of antimicrobials for animal production and even for pets. There is no need yet in Brazil for a prescription to buy antimicrobials. So people don't need to get a prescription, a veterinary prescription, to buy antimicrobials for pets or as well for production animals. The only thing we have changed a little bit in the few years was that we have some restrictions about the use of antimicrobial as growth promoters. We have a list of antimicrobials that can be used for that, not all antimicrobials. The Minister of Agriculture nowadays are trying to restrict some drugs, some bases just for use in humans and some just for use in animals. But for the treatment of disease, like for pets and even for cows, we do not need a prescription to buy antimicrobials. And also, we do not see a very nice database, a central database for the animal health authorities that we can use to see which are the main drugs used in Brazil and for which animals they are used, like which is the main drug used for pigs or for cattle in Brazil. We don't know how many tons are used per year and which one are used for treatment or which one are used for prophylaxis or metaphylaxis treatment. And so we do not have this official and common database that we could use to understand the behavior of the use and consequently the resistance in Brazil and the impact of the animal production in the antimicrobial resistance. So we don't know what is used, how much, and how and in which species, we don't know. So the first, in my opinion, the first step that we should take in Brazil is to understand better our problem, how big it is, and how it's the size of this problem in Brazil. And I believe also that the university and the researchers, they have a central role in this One Health approach and in this antimicrobial use and antimicrobial resistance, because we have the power or the source to understand the better scientific evidence better than the other players in the milk chain or in the animal production chain. And also, we do not have some conflict of interest that here in Brazil, I don't know how it is in US, but sometimes when we sit together to talk with people from industry, from farms, because in Brazil also we have some industries that are just responsible for buying the milk from the producers and sell to the industry. So we have like these people in the middle. And when we sit all together, we feel sometimes that people think that they are in the opposite sides of the problem. Farmers, they blame the industry for having been more restricted about the antimicrobial residues in the milk, and also people from the industry, they blame the farmers, and sometimes it's difficult to be in the middle. But I think the university, with the credibility that it has, and with the researchers that are in the front, on the frontier of the knowledge, we can have this central point, and also trying to help people that our animal health authorities who make policies in this way, in agriculture side as well in the human health side, help these people to find how is the middle way, how can we reduce the use of antimicrobial in dairy farm as well as in beef farms, and get more animals health, and also people in the animal products of higher quality without risks for population. And so it's my feeling, I think we have to talk all together and make people have an agreement about how we are going to address this problem. So thinking in this, we have started about 1.5 years ago this project that is called Quantification and Classification of the Risks Associated with the Use of Antimicrobials on Dairy Farm in Minas Gerais. This is a project that have collaboration of people in the industry, farmers, and some universities in Minas Gerais. So our goals are identify the main antimicrobials used in dairy farm in Minas Gerais, because we don't have this information from the animal health authorities in Brazil, because antimicrobials can be sold without prescriptions. So we also want to evaluate for which therapeutic protocols these antimicrobials are used, which are the costs. This is something that also I think every time I talk to farmers, I usually try to get them to use less antimicrobials because of the costs. And it's not difficult to show to them that if they have a better records of the treated animals and better records of everything, they can diminish, they can reduce the cost associated with antimicrobial use, so they have better profitability of the activity, the animal production. So we are trying to understand for which disease they are using antimicrobial, and if the doses and the treatment are proper, are correct. So we are, in the end of this project, we are going to have an individual report for each farmer who participated of the study, so we can help them to address the problems that they have in the farm. Also, we are going to isolate some strains of the farm, and trying to understand if the resistant profile of these strains are or not associated with the use of antimicrobials in that farm. And also, we are going to carry on extension activities individually, directly for tailored for each farmer, trying to help them improve the use of antimicrobial in their farms. And also, in the end, we are planning to have a document, a report that we are going to send to the public policy makers in Brazil, so they could use this information to design other policies in Brazil for control the use of antimicrobials in animal production. This is a joint project, us at UFLA and UFMG, that is another university in Minas Gerais, the Federal University of Minas Gerais, in Belo Horizonte, which is the capital, the capital of the state. Also in Brapa, that is a public enterprise that it's dedicated to research, and CCPR, that is one of the main players in Minas Gerais, actually is the main player that buys the milk from the producers and sell to the industry. This company is a big company in Minas Gerais. They per month they buy around 9 million liters of milk and sell to the industry. So when we start this project, we have 176 dairy farms, all in Minas Gerais. Altogether, these farms, they have around 13,000 dairy cows. And we have a project that was supposed to follow up these farms for 12 months. each 3 months, we go to the farm and we get the information of all antimicrobials that the farmer use or their employers used for treatment of all animal categories, calves and cows. We use the two methodologies. Each treatment should be registered in a form, and also each bottle of antimicrobial that was used and ended was disposed in a trash that was exclusively used for the proposal of this study. So we have both ways to understand which were the antimicrobials that the farmer were using most. Which the records from the form, we could say which disease were treated, but we think we have a better record of which drugs were used by the trash methodology. So every 3 months, we go to the farm, we collect the bottles and also the forms, and we analyze the data, and we are trying to understand which are the profile of use of antimicrobial in each dairy farm in Minas Gerais that are participants of this study. This is the project, the people going to the farm. Here are some of the students that are participating in this project, and the producers also. And here are the distribution of the farms in Minas Gerais. We could not expand the project for the whole state because our state is really big. I don't know if you are familiar, but Minas Gerais state is almost the same size of France, Italy, and Belgium together. So we are a big state, and we also have a lot of farms. We are the main state in number of farms also in Brazil. So we have this sampling that was possible to carry on the follow-up of 2022, the 12 months. So this is the trash, the garbage that we put in the farm. So the farmer should put all the bottles of antimicrobials in this box. So we collect them frequently. And also we put there a notebook as this one here. So he could, the farmer could put all the records, all the treatment that were done, and all after the collection of this notebook, they were photographed and put in the Google form that we use to analyze the data. This is the collection. People in the lab were counting all the bottles and also the intramammary antimicrobials. And we finished this period actually in August this year. We finished the follow-up of 12 months of the all farms. And we are now trying to complete and consolidate and organize the whole database. The counting of the antimicrobials, we finished it. We are now just cleaning the database because all the forms that were in the hand notes, they have to be put in the Excel tables and then analyzed in R. So this is the part that we are doing right now. But I have some results from the first quarterly of the first 3 months of the follow-up. I'm going to show you. This is just to show you that most of the results, we are going to focus on the mastitis antimicrobials because this is not from Brazil, this is a study from Canada, but they show that around 70% of the antimicrobial that are used in a dairy farm, it goes for treatment of mastitis, clinical mastitis or dry cow therapy. So some of the results I'm going to show you just for mastitis. I'm going to show the overview of the antimicrobial use, but some of them I'm going to have a special attention in mastitis because we have this data from the literature that shows that most of the antimicrobials in a dairy farm goes to the mastitis treatment. What we have done. besides the collection of the antimicrobial use information in the dairy farms using two methods, the form and the bean method, we also took a sample, the bulk milk sample of each of those farms. We send it to the lab. We select until 5 columns with different morphologies from that milk sample. And we identify these 5 columns when we could recover the 5 columns from the sample by Maud-Toffee. And we did some MICs, minimal inhibitory concentration, to understand the profile of antimicrobial resistance in these strains, isolated from the milk samples. And also, we are going to select 15 farms to do shotgun metagenomic in this milk, the samples of milk, to try to understand if the profile of antimicrobial use that we are seeing are or not associated with the profile of antimicrobial resistance. So then for the antimicrobial resistance, we are going to have 2 different ways to look at it. The one is going to be the strains that we have isolated from the milk samples, the bulk milk samples. And also we are going to look at the genetic profile of resistance by doing the shotgun. We wish we could have more a bigger budget, so we could do the shotgun metagenomic in all farms. But as we have restricted budget, we're going to do just in the 15 that have the better record of antimicrobial use in the farm. So we are going to select these 15 farms that are going to have the metagenomic doing based on the better adherence of to the project. So our preliminary results show that we have in the beginning 171 farms, 176, I'm sorry, but just 171 responded the initial questionnaire. This initial questionnaire was for understand the profile of the farm, how many cows in lactation, how many dry cows, how many cows, and which was the system of milking, all these questions that were general. So not all farms responded the questionnaire. And the follow-up ended with 105 farms only. Several farms, they dropped the study for different reasons. Some because they stopped selling the milk for the CCPR, some because they felt that it was difficult to record the use of antimicrobials, some because they just did not put the bottle of antimicrobial in the trash that they should. Different reasons. Some farmers they abandoned the activity. So we ended up with 105 farms. In the first quarterly, in the first 30 months, we have 1,108 treatments registered, and from the milk samples that we took from the bulk milk tank, we got 185 isolates. So talking about the dairy farms profile that started or ended the study, we have different milk production daily. We have most of the farms producing around 200 to 1,000 liters of milk per day, but we also have very small farms. This is something very particular from Minas Gerais state. From other states in Brazil, we have bigger farms, but here we have a tradition that very small farms, and farms as with like 50 cows, lactating cows. So we have this particular characteristic of Minas Gerais. So we use the wasted bean method and also the forms. We have ended up the 12 months with 105 farms, and I'm going to show you some of the results. when we asked for the producer what they do with the milk with antimicrobial residues, 55% say that they used to give to calves. So we think that these maybe have an impact on the microbiome of these calves. Also, we have the most of the farms using pre and post dip for treatment for prevention of mastitis. And most of the farms use mechanical milking. It may be surprising you, but in Brazil we still have, especially in Minas Gerais, where we have small farms, we still have farms that use manual milking. But composing the study, most of the farms use mechanical milking, and 74% of the farms exclusively produce milk, not having beef or other activities in the farms. And 70% of them, the farmers, they use antimicrobials without veterinary guidance. So this is something that we have to pay attention. Most of the farms, they use antimicrobials following their own head. They do not ask any veterinarian about the use of the antimicrobials. So the results from the first 3 months show us that the leading antimicrobial use was this one, cephalosporin. Most of the most used antimicrobial was cephalosporin. We have the rank here, and most of them were used to treat mastitis, as we see in the study of Canada. Here, mastitis is the first one. 43% of the antimicrobials used in the farms were for treatment of clinical mastitis. For dry cow therapy, we have like 80.77% of the antimicrobials used. We registered more than 1,000 treatments in the records of the farms. We have here the whole set of the disease that were treated with antimicrobials in the farms. When we divide the antimicrobial use considering the production of the farms, we see that bigger farms use more antimicrobials than the small farms. But after 200 liters per day, there is no much difference between the farms. Here we see the main drugs used. When we see tetracycline and cephalosporin, this yellow one and the light blue one were the most used independently of the production of the farm. Most of the farm use these bases, and also penicillin and associated with aminoglycoside. The profile of use of the base used did not change very much according to the production. When we consider treatment of mastitis, only not all the whole disease that can be treated with antimicrobials, we see a difference more than considering the whole budget of disease, but it's still not too much different between the production strata. Here is something that we have to be very concerned. Most of the antimicrobials used in the farm are critically or highly important for human population. Most of them, just one is not considered by World Health Organization as critically or highly important for the human population. So this is something that calls attention because this is used for mastitis, the ones that can be maybe found in the milk that is consumed by children and elderly people. So this is something that I was especially concerned about. Most of the antimicrobials used to treat mastitis are critically or highly important for human population. And we also have performed the MICs for the strains that we have isolated from the bulk milk tank. Here is the panel of strains that we have isolated. We isolated several species of bacteria from the milk. Staphylococcus aureus was the mainly one isolated, but we have also other strains that are species that are not associated directly with mastitis. For the ones that we see direct association with mastitis, Staphylococcus aureus, and Streptococcus agalacti, we have performed the MICs tests. We choose the antimicrobials that are most used in the farm and also the ones that are used for treat human disease. These results I do not have to share with you. We are finishing the MICs. I think next week we are going to finish all the analysis in the laboratory. But we still have to finish the MICs for Streptococcus aureus and Staphylococcus aureus and Streptococcus agalacti. We are going to analyze the data because we have put all in the spreadsheets, Excel spreadsheets, and we are now putting in R to analyze the prescription of the antimicrobials used in the 12 months follow-up, and also the trash that we collect from the farms. And we are preparing the individual report for the producers. We have a meeting settled with the producers for January. We are going to have a collective meeting. We are going to share the results, the project with the producers, and then we are going to plan individual visits with the students leading that to each farm that participated, the student, and giving the farmer the report, the individual report, saying what they should change in the daily basis for changing the antimicrobial use and the antimicrobial resistance. And also we have performed the selection of the farms that are going to be sampled for the shotgun metagenomics. We have this sampling schedule for December. And we also are planning to present these results for the Animal Health Authority at the Ministry of Agriculture here in Brazil. And we expect the benefits from this project that we think it's a One Health approach, because we are talking to different actors from this chain. We expect to contribute to implementation of regulatory measures in Brazil, showing the animal health authorities that we have a problem. We expect to find some association between the use and antimicrobial resistance in the data that we have. We are thinking that the project may maybe contribute to the animal health and also for the human health, because we believe in the use of antimicrobial, but we also believe that it should be used responsible, and the use should be responsible and for only when is necessary, strictly necessary. And we also think that this initiative can straighten the relationship and reduce the, how can I say, the difficulties that people from industry, from farms have to talk. I think we can put all people together to try to develop together a solution, a midterm solution for everyone. And also, I truly believe that farmers, they sometimes use antimicrobials because they don't know how is the best way to use it. They use it in the wrong way because they don't have the knowledge. So trying to personalize, to individualize the results of the project, we can help people to make a better use of the antimicrobes that they have. And also with that, they can reduce the cost of the production and have more profitability of the activity. And also the industry, they claim that they have to spend millions of reals every year with the incineration of the meat, the cremation of milk. So if we reduce the milk that must be sent to cremation, we are also improving the quality of the milk and the international competitiveness of the milk produced in Minas Gerais, and also the profitability of the industry. So we believe the information is power, and information should be shared with every people in the chain, the industry, animal health authority, farmers, everyone who is involved in the milk production. So as the final message, I think only well-managed farms can reduce the antimicrobial use or antibiotic use. So we should give the farmer the power of deciding better if they should or not treat an animal and the way they should treat this animal. And we have to establish action plans that involve everyone in the chain, all people who participate in the dairy production in Brazil. And we have to find a way to quantify the use of antimicrobial and to do the surveillance of antimicrobial resistance. I think it's a need, urgent need in Brazil. We are very late in doing this kind of action. We are the 50 in the global rank of milk production. So we must do that. And I think One Health approach, it's fundamental in this process. It's the only way, in my opinion, to get good results that will benefit everyone, including the consumers. So I want to thank all people who are involved in this project. I mentioned that the UFMG and UFLA and Embrapa and CCPR, but we have also the institutions in Minas Gerais in Brazil that are funding our project and also are contributing in other ways. We have people from Calgary University, Dr. Herman Bakema, and Professor Javier Sanchez in the UPI who are helping us with the analysis. And also from other companies as Cia do Leite Laboratory and the Cia do Leite Consultory who are helping us to understand if our results make sense for the producers. And also in this contact with the producers. And also I want to thank all the people in the laboratory who are with us in the field as well as in the lab, and also my family that help to be out of home several times. And also I want to thank you for the attention, and I'm open to questions, and I'm really honored to be here talking for a selective audience, and it's a very pleasure for me. And sorry for my English, it's been a long time since I practiced, so it's not something that I talk every day, and so I apologize. Thank you.
Elaine, thank you so much. Appreciate your English is really good. So you should have no apology necessary for that. That was wonderful. Thank you for sharing with us all. I want to remind everyone that the chat is open for questions, and I might pass back over to Dr. Miller if she wants to handle and coordinate questions for you,
Sure.
Elaine.
Absolutely. Absolutely. I'm going to start out though. You will be coming here and you can hear me, right?
Yes, I can.
Okay, awesome. You'll be coming here again in the spring. And this was an awesome presentation that you just gave. But I am curious, some of the things you're going to want to do in the spring is to explore some collaborative opportunities. Do you have any thoughts right now that you could share with the group so that people who might not be aware of your visit in the spring, but it might get them to put it on their calendar?
Okay, thank you. What I think we could do in collaboration, we have big bank of strains. So we have, because in our laboratory, we also offer to farmers some services, like they can have a microbiological diagnosis of mastitis, sending samples from our laboratory. So we have a huge amount of strains isolated from milk. So maybe we can do something together, sequencing them or trying to understand from chronological way, because we have older strains and new strains, if we have change in the profile of resistance. Together with these strains, we also have a database that say where these strains were isolated, which farm, and if it was from bulk tank milk or individual cows. We also have way to analyze the data of sequence. You have a bioinformaticians in the lab, so we can do something together. We also have here access very easily to the farms, so we can develop a new entire project together. And also, I work also in brucellosis and tuberculosis. I don't know if there is anyone who is interested in this other disease, because we are also developing some new tests that can be evaluated using different samples, serum samples, for example. I think we have several projects that we can develop together, I'm pretty sure. And also we have a very easy access to the productive sector here in Brazil, something that may be of interest from people there.
Excellent. I have another question, because I know we have some people on who are experts in like watersheds and water facilities. So I'm curious, have you, when you're doing your study and some of the other studies that you've done, whether it's looking at antimicrobial resistance or specific pathogens, are you also looking at, because in some cases they look at watersheds and the ground and what's being put into fields and things like that. Do you look at that broader area, I guess?
Not in this project, but we have another project that is going on now. I haven't told you, but Minas Gerais is not only the most important country in the milk production, but also in the artisanal cheese. Here in our state, we produce a very special cheese that is very cultural thing, and some of the identity of the people here, that is made from raw milk. So this is problem, because not only from the antimicrobial resistance perspective, but also from the zoonotic importance of some bacteria. And then in this project, we are looking at bacteria in the place where the milk, the cheese is ripening, in the water from the place where the milk is produced, the cheese is produced, in the milk, and also in the farm, in the animals in the farm. So we have a bigger project there. We are doing this project in Minas Gerais. We have 10 regions that can produce artisanal cheese. They are recognized by the UNESCO as some immaterial heritage from Minas Gerais. So we collect samples during the ripening process in 3 different regions in Minas Gerais. So in this project, we are seeing in a broad way the resistance in the environment, in the animals, and in the cheese, and in the milk that you use to produce the cheese. So we are looking in a broader view, the antimicrobial resistance and how it can cross these barriers in the environment, in the animal, and in the project.
That's very interesting. I know even here there's a big push for raw milk. And I grew up on a dairy farm,
Yes,
so I grew up on raw milk. But we knew on the cow that was giving us the raw milk.
that's
And
the
it
problem.
is, yeah, and it's very concerning to me that there is that, that push without having consideration for all these things that we started pasteurizing milk for. But at the same point in time, I understand the value of raw milk as well.
Yes, for us in Minas Gerais, it's something very cultural. It's the identity of the
Yeah,
people here. So, and for the legislation, we cannot pasteurize this milk.
right.
But we still have problem with brucellosis and mastitis and tuberculosis and leptospirosis and so on. So I think we should have other ways to address this problem here. So we are trying to understand this. How can this antimicrobial resistance and bacterial zoonotic bacterial disease can spread until the project?
Excellent. So you mentioned leptospirosis also. So I know some of our colleagues were not able to be on the call today from the vet school because there's a interview going on, but one is very interested in leptospirosis, and so
Oh, good.
yeah, so that's is of great interest. Is that something that one of the pathogens
Yes.
that okay,
Yeah, we
yeah.
work with leptospirosis too. I do not test in our lab here, but I have a collaboration with USP, University from São Paulo. People there, we have all samples we collect, we test for leptospirosis, and actually we are seeing a very different profile leptospirosis here in Brazil. They causing more reproductive losses in the first third of the pregnancy, not abortions, just some kind of endometrites. And also we see no systemic antibodies being produced, but PCR positive in the uterus of the animal. So we have interested, and we have some projects on leptospirosis as well.
Wonderful. Tom, do you want to ask anything, or does anybody have any
We,
questions?
we have a question from Benti,
Perfect.
but I've asked if they can post it in the chat. It was just sent direct to me. But I
Okay.
think so that it goes in the chat so I don't mispronounce things.
Benti, if you want to unmute yourself and ask, I think.
Sure. Thank you so much for
There
the
you go.
nice presentation. So I joined the presentation very late, almost after 25 minutes. So I apologize if I missed what I'm going to ask you. So given the high use of third and fourth generation cephalosporins, is there any information about the current status of extended spectrum beta-lactamase producing bacteria in the dairy system in Brazil?
We are going to do that in this project. We are planning after the MITs running the searching for these enzymes, but we haven't done yet because we just finished the isolations, and we are finishing the MICs next week. So when I go in the spring, I probably will have this, but I believe yes, because we have another project that one of my PhD students, she found a very concerning, alarming number of strains SPL producing in the, but it was in E. coli from calves with diarrhea. But I believe it's the same because they use the milk with antimicrobial for feed the calves in Brazil usually.
Okay, thank you so much. I think it would also be interesting if we have also the same issue here. There is high prevalence of extended spectrum beta-lactamase producing, especially among Enterobacteriaceae. And again,
Yes.
we don't use fourth generation cephalosporin here. Third generation cephalosporin, special safety of is common here in the US. So do you have also a plan to look at, because here from the US side, if you see the CDC report is indicating that there is a continuous rise in extended spectrum beta-lactamase producing Enterobacteriaceae. The infection rate is increasing and is mostly community associated. It's used to be normally a kind of hospital acquired. So although there is no direct link, that we don't have evidence about direct link between transmission from animal to human, and it's also not easy to look at that link here. So do you have a plan to look potential pathogen sharing between at least dairy workers and also the dairy animals? Yeah.
Not in this project because we haven't collected the sample. Actually, collecting samples from dairy workers, I believe it's difficult also in US. Here it's very difficult because of ethical issues. So we haven't done that in this project. But in the project with cheese, we are planning to do that because it would be very nice if you have to look at the cheese, the environment, and the cow, and also in the workers that are in the cheese making process. But in this particular product I presented today, no, because the samples were already collected. In the isolates from the Enterobacteriaceae family, we are going to look at the ESBL producing strains. And also in the shotgun that we are going to do, we are planning to look at the genes related to the ESBL production. And unfortunately, in Brazil, we can use the 4th generation cephalosporin for treat animals.
Yeah, thank you so much.
Thank you.
We have time for another question if anybody has one. While we're waiting, I do want to know, because this just come up, when they're feeding the waste milk to the calves, are they mixing it with milk replacer as well?
It depends. Usually, it depends, because if there is sufficient cows in the treatment of mastitis, they don't need to. But usually, yes, they mixture, but not always, not all farms. It depends of the prevalence of clinical mastitis at the time. But actually here, there is a plan, but it's not in place, that some truck should go to the farm and collect all this waste milk, but that is not a reality today.
Right. Any other questions? All right. Well, I guess we're almost at the hour anyway. So thank you again. I really, really appreciate that.
Thank you. Thank you for having me, for the invitation. I'm honored.
Thank you. And thank you, Tom, for making our connection.
Yeah, thanks, Deb. Thanks, Elaine.
Thank you, Tom.
Goodbye.
Thank you, everybody.
Be in touch.
Bye.
Bye.
Bye-bye.